Okay everyone, welcome back for our next lecture video. I'm very excited for this lecture video because this is one of my favorite topics in all of anatomy and physiology. This video will cover chapter 10, which is muscular tissue. So in this class we're going to talk about how muscles work, how muscles contract. We're going to talk about the structure of muscle and the different concepts behind what makes muscle tissue so very special. So think back to our tissues chapter. Muscles, in fact, are their own type of tissue. And this type of tissue is specialized to contract. There's going to be certain organelles inside of our cells that allow for muscle contraction. And muscle contraction is going to be one of the major concepts that we're going to learn in this video. So as a brief introduction, we're going to learn the three different types of muscles and how they're structured and where they're found. Our three types of muscles are skeletal muscle, smooth muscle, and cardiac muscle. And each type of muscle is going to have its own unique characteristics, which help it to function in the part of the body that it's located. We're also going to introduce something known as the neuromuscular junction. This is the location by which nerve cells are going to attach to muscle cells, and that's going to facilitate the contraction. Whenever you want to contract one of your muscles, the brain is going to send out signals to that muscle and tell that muscle to shorten in length. And the muscle contraction is what's facilitated at this neuromuscular junction. So we'll talk about what events happen at this location as well. We're also going to discuss the contraction cycle and how energy can be used in our muscle cells to produce a muscle contraction. We're going to go over the entire process by which nerve cells will send out signals to muscle cells, and we'll get a muscle contraction all the way until our muscles become relaxed. So chapter 10 is very information rich, so I'm going to go ahead and get right into this and start with the properties of muscular tissue as a whole. So while all muscular tissue is specialized for muscle contraction, there are some other properties that our muscle tissue will have as well, namely electrical excitability, contractility, extensibility, and elasticity. Now we're aware that muscle cells contract. That's what contractility means. The ability for our muscles to shorten in length and to produce some type of tension or force. But there are in fact some other properties as well, so let's take some time to go over each of these. So the second of our properties of muscular tissue is electrical excitability. This means that certain cells are able to produce action potentials. Now, I've only briefly mentioned the action potential a few times up until this point. When we get to chapter 12, we're going to discuss action potentials in much greater detail. But for now, I just want you to understand that action potentials are signals that can be sent from cell to cell. So certain types of muscular tissue are going to have this electrical excitability, and they're going to be able to generate action potentials and send signals to other muscle cells all on their own. One of the best examples of this is cardiac muscle. Cardiac muscle has certain cells that allow it to contract all on its own. Our next major property of muscular tissue is extensibility. Extensibility refers to our muscles' ability to stretch within reason. As there are muscles found all over our body, our muscles are going to need the ability to stretch without being damaged. So that's what extensibility refers to. And lastly is elasticity. And elasticity refers to the ability for our muscles to regain their normal shape after they have been stretched. So muscle tissue is very, very resilient. It has the ability to contract, it has the ability to produce its own signals, but it also has the ability to stretch within reason without being damaged. So the properties of our muscular tissue are going to indicate the functions of our muscular tissue as well. So skeletal muscle attaches to the bones. That's where our skeletal muscle is going to attach to. And that is the type of muscle that's going to help us to produce useful body movements in conjunction with the skeletal system. Our skeletal muscles are also going to help us to maintain posture as well as stabilize our body. I've mentioned frequently throughout these lecture videos that the muscular system and the skeletal system work hand in hand to produce useful body movements. Without these two systems working together, our bodies would not be able to move in the ways that they do. So the skeletal system gives us surface area for our muscles to attach to through tendons. And our muscles themselves are what do the contraction to pivot our bones in certain directions to give us these body movements and stabilize our body positions. Now, cardiac muscle and smooth muscle are also going to be doing a lot of this function within the body. So our hollow organs are going to store and move substances throughout our body. Think about cardiac muscle. Cardiac muscle makes up the walls of the heart, and the heart is functioning as a pump. The heart pumps blood through the vascular system. And so it is the muscular tissue of the walls of the heart that pump the blood through the blood vessels. Smooth muscle is going to control the movement of food through the digestive system. Muscles in the esophagus, the stomach, the small intestine, and the large intestine are all going to contract to move things through the digestive system, and that's going to help to move the food around so that we can absorb it in the right places and then eventually release it. So while we often think of the muscular system as skeletal muscles, we have to always remember that smooth muscle and cardiac muscle also play a role in moving substances throughout our bodies. Our last major function of the muscular system is heat generation. When muscles contract, they undergo exergonic reactions that will release heat. And this is why when we exercise, our body temperature goes up and we will sweat. So these are our 4 major functions of the muscular system. Each function is vital for our body's homeostasis in different ways. So on this slide, we're going to describe the 3 types of muscles. We're going to discuss where they can be found and also what their appearance is. But before I go into the 3 different types of muscles, I just want to take a second to explain muscle cells. So all 3 of these types of muscles look a little bit different, but all muscle cells can also be referred to as muscle fibers. Muscle cells are often these long cylindrically shaped cells, and under the microscope, they represent a long fiber. So that's why we refer to muscle cells as muscle fibers. I will often be using this term throughout this presentation, but whenever I say muscle fiber or refer to something as a muscle fiber, I'm just talking about a single muscle cell. And again, the reason that we call these muscle fibers is because they are long and cylindrical, just as fibers are. So skeletal muscle fibers, cardiac muscle fibers, and smooth muscle fibers are all going to appear slightly different. There are lots of similarities between these 3 types of muscle tissue, but each type is different in its own way. So as I've already explained, skeletal muscle can be found attaching to the bones. Skeletal muscle is named because it attaches to the skeleton. Cardiac muscle can be found in the heart, hence its name, and smooth muscle, which can also be referred to as visceral muscle, can be found in the walls of our hollow organs of the GI tract, as well as the urinary bladder. So each of these types of muscles is named based on where it's found. And all muscle is going to have the same properties, so all muscle contracts. But based on where the contraction occurs is where we get the function. So our skeletal muscle, of course, is going to help to move our bones around, and this is what gives our bodies useful movement. Cardiac muscle is going to pump blood through the blood vessels of our bodies. And smooth muscle is going to move other substances through our body. Again, this might move food in the digestive system or perhaps urine in the urinary bladder. Now, skeletal muscle is our only type of voluntary muscle. This is the only type of muscle that we get to consciously control. Cardiac muscle and smooth muscle are both involuntary types of muscle. We could not consciously contract those muscles even if we wanted to. And this is a good thing as well. The heart pumps roughly 1 time every second for our entire lives. And it would be very, very inefficient if every time our heart beat, we had to think about beating our heart. So it's very nice that this has involuntary muscle action. Smooth muscle as well is going to be an involuntary type of muscle. So skeletal muscle is our only type of muscle that allows for us to have voluntary control. Now lastly for this is how these cells appear. Under the microscope, each of these types of tissues are distinguishable and they have different characteristics. Skeletal muscles will have multinuclei and will have a structure known as striations. Striations refer to the stripes. Now this is a muscle fiber that's coursing from left to right. The striations will always run perpendicular to the fiber length. So you can see these alternating light and dark bands or stripes on our skeletal muscle fiber. That is showing us striations. Skeletal muscles will also have lots of different peripherally located nuclei. These are just nuclei that are found on the sides of the cell. So that's what skeletal muscle is going to look like under the microscope. That is its appearance. Cardiac muscle has some similarities, but it also has some differences as well. Cardiac muscle is going to also have striations, so we'll see these nice stripes, these alternating light and dark bands coming across our cardiac muscles. However, cardiac muscle cells only have a single nuclei, and the cell shape is no longer cylindrical. It is actually going to be a branched shaped cell. So these cells, attaching to one another, are going to form a network of muscle tissue. Now thinking about where these muscles are found and what their function is going to be, it makes a lot of sense that skeletal muscles are a long cylinder and cardiac muscle cells branch to form a network. Skeletal muscle fibers are overall shortening the length of an entire muscle. So it makes sense that we would have a long cylinder that's going to shorten in one direction because our muscle is going to be in mostly one direction. Cardiac muscle is a little different. Cardiac muscle, found in the walls of the heart, need to contract around a chamber. So it makes sense that these muscle cells are going to have this branched arrangement because that allows them to form a network of muscle fibers that will contract around a chamber. The most characteristic structure that we will find in cardiac muscle cells are the intercalated discs. The intercalated discs are the little junctions that can be found between more than one muscle fiber. So these muscle cells will bind to other muscle cells in branched formations. And at the location of binding, we will find the intercalated discs. Smooth muscle, on the other hand, is going to be the one type of muscle that does not have striations. So both skeletal muscle and cardiac muscle will both have striations, smooth muscle will not. And that's based on the different arrangements inside of the smooth muscle cell. Smooth muscle will also have a single centrally located nuclei, and the ends of our smooth muscle cells will taper. You can see that we'll have a plump center and two skinny ends. Oftentimes, smooth muscle cells are referred to by histologists as cigar-shaped, and that is based on this plump center and the two skinny ends. So these will characterize the function, location, appearance, and control of our three different types of muscles. I want you to feel very comfortable with distinguishing these types of muscles based on the criteria laid out on this slide. So all muscles produce some type of movement, and I discussed in Chapter 9 a little bit about how this works. So all muscles attach to bones through structures known as tendons, and tendons are a type of dense, regular connective tissue. So when muscles contract and shorten in length, they're going to be attached to two different bones. And so as our muscles contract, that's going to pull our bones in one direction. So while the muscle contracts, that's going to pull on the tendon, which will eventually pull on the bone, and that's how we get movement. Now there's one other term that I'd like you to understand, and that is what is known as an aponeurosis. Aponeuroses are just sheet-like tendons that are very, very flat. Oftentimes, tendons are more cord-shaped and have a cylindrical structure, but when they are flat-shaped and very thin, we call those aponeuroses. We'll find certain examples of aponeuroses throughout the body, but I just want you to understand that the difference in the name is just based on the difference in the structure of the exact tendon we're talking about. So as I just mentioned, all muscles attach to different bones at more than one place, and the places by which muscles attach to bones get their own name. So this is going to bring us into the concept of origin and insertion. The origin of a muscle is the point on that muscle that attaches to the immovable bone when undergoing some type of movement. So most often, muscles will cross 1 to 2 joints, and this will allow the facilitation of movements and actually changing the angle between those articulating bones. So you can see that the biceps brachii muscle is actually spanning not only one joint up here at the shoulder, but also another joint here down at the elbow. And when this muscle contracts, that's going to allow movement at 2 different joints. And that is why muscles will often span more than one joint. When the muscle contracts, you can imagine if this biceps brachii muscle was to contract, that's going to end up pulling this bone up like so, and it's going to move our bone and decrease the angle between these two articulating bones. So every skeletal muscle must attach to the skeleton, and the specific places that our muscles are attaching to our bones is going to determine the action of that muscle. So the attachment points can be referred to as origin and insertion. The origin of a muscle is the attachment point to the bone that remains stationary throughout the movement. So for the biceps brachii muscle, our origin is going to be found up here because this is going to be the bone that moves when this muscle contracts. So the origin is the attachment to the bone that does not move. The insertion, on the other hand, is the attachment point to the bone that does move. So this is going to be the insertion of the biceps brachii muscle. When this muscle contracts, this bone moves. And since this attachment is on the radius bone, this is going to be the insertion of this muscle. Each muscle in the body is going to have its own origins and insertions. Some will have more than one origin, some will have more than one insertions. It just depends on the muscle that we're looking at. But what will always remain the same is the definition of origin and insertion. The origin is always going to attach to the bone that does not move. The insertion will always attach to the bone that does move. Now, most of our body movements are not facilitated by just one muscle. They are actually facilitated with groups of muscles and certain muscles that actually oppose one another. So muscle movements are often coordinated by multiple different muscles in one group. And so when we refer to different muscle movements, we have a few other terms that we're going to use to describe the action of different muscles during given movements. So our first term is an agonist, or a prime mover. This is any muscle that contributes the majority of muscle contraction to any movement. So if I'm standing in the anatomical position and I want to bring my hand up to my shoulder, I'm going to contract my biceps brachii muscle, which was the muscle that we saw right here on this slide. If I'm trying to bring my hand up towards my shoulder, I'm going to contract this biceps brachii muscle. But this is not the only muscle that gets contracted during this movement. There are other muscles that play a role in stabilizing and also opposing this movement. However, the biceps brachii muscle is going to do most of the work in pulling my hand up to my shoulder. So therefore, the biceps brachii in this movement would be referred to as the agonist or the prime mover. Now, agonist muscles will also have oppositional muscles, and those are known as antagonists. Antagonists are muscles that oppose the action of the agonist. Now, this would seem sort of counterintuitive. Why would we want a muscle to oppose the muscle movement that we're trying to make? Well, because these muscles help to control the movement. If I did not have the antagonists, my prime mover would contract more forcefully, and that would give me uncontrolled movements. So a great example of an antagonist is the triceps brachii, which is found on the back side of the arm. You can see that this muscle also is going to span more than one joint, and it's going to be the antagonist to the biceps brachii muscle. So when this muscle contracts and shortens the entire length of this muscle and pulls the radius in the hand up towards the shoulder, the triceps is also contracting very slightly, and this is helping to produce a smooth overall muscle movement. Antagonists will often be found on the other side of the bone than our agonist is, and again, these types of muscles are always going to help control the action of the agonist during a particular muscle movement. Now we'll also have synergists. Synergists are going to aid in the muscle action of the agonist by helping to stabilize the particular joints that are moving. And for most of our types of muscle actions, we're going to see all three of these types of muscles included in any given muscle movement. So if I now want to take my hand back to the anatomical position, the agonist is now going to become the triceps brachii, and the antagonist is now going to be the biceps brachii. The definitions of these words solely depend on the muscle movement that is being facilitated. And almost all of our muscle movements are going to be facilitated by the coordination of muscle groups and more than one muscle rather than just one muscle on its own. So next we're going to discuss some of the criteria that we will often use to name the specific muscles that we find in our bodies. Now there are lots of different criteria that we use, and that's how we get so many unique names for our muscles. So let's go over the criteria for these. First and foremost is the location of the muscle. The location of a muscle will often be something that we use in the name of the muscle, and this will help us when we hear that muscle name to automatically know where that muscle can be located. Certain different muscles might be found in the same area, but they might be bigger or smaller than each other, so we will also use the size of the muscle to denote the names of these muscles as well. The number of origins, as well as the location of the origin and insertion, so the muscle attachment points can also play a major role in how we name muscles. The fiber direction, as well as the appearance, are two other criteria that we can use. Certain muscles are simply named based on the way that they look, and muscles will also be named based on the actions that they provide for the body. So flexor and extensor muscles on the forearm are going to indicate the action of those groups of muscles. So let's go over each one of these specifically, and I'll give you an example of where we will use this naming criteria in the body. So first and foremost is the location of the muscle. The temporalis muscle is found on the outside of the temporal bone. This muscle is solely named based on where it is located on the body. And we'll see a lot more examples as we go through the body and learn different muscles. But especially in your lab class, when you're actually going to learn the specific muscles of the body, you will see the location of the muscle used in the name very frequently. The size of the muscle is our next criteria. We have the gluteus maximus muscle, we have the gluteus medius muscle, and we have the gluteus minimus muscle. All of these are located in the gluteal region, however, one is larger than the others. And we call that muscle the gluteus maximus. So this is an example of using both location and size to name a muscle. The number of origins is another criteria that we've used. We've already briefly talked about the triceps brachii and the biceps brachii. Tri stands for 3, so triceps brachii is going to have 3 origins on it. Bi stands for 2, so biceps brachii is going to have 2 origins on it. Next, we will talk about appearance. The appearance of our muscles is something that we will also use. The trapezius muscle sort of resembles a trapezoid, so that's another criteria that we might use. We also may use the direction of the fiber length. So for an example, the rectus abdominis. Rectus means straight and helps us to stand erect. The rectus abdominis is the muscle that makes up the abs. So that's the muscle that we're always trying to hone before summertime. So the rectus abdominis is named based on the direction of the fibers that can be found in that muscle. We'll also see the location of the attachment points playing a major role here. So a good example of this is in the sternocleidomastoid muscle. The sternocleidomastoid muscle is a muscle that can be found in the neck that attaches to the mastoid process of the skull and the sternum as well as the clavicle. So we get sterno for sternum, cleido for clavicle, and mastoid for mastoid process. So this muscle's name is telling you exactly where the origins and insertions of this muscle can be found. And lastly is the muscle action, which are the flexors and the extensors. We will see flexor muscles in the anterior compartment of the forearm in the anatomical position, and we'll see extensor muscles found in the posterior compartment of the forearm. So these are the major criteria that we use to name our muscles: location, size, the number of origins, as well as the location of the origin and the insertion, the appearance, the direction of the fiber arrangement, and the actions of the muscles that we're trying to name. So that will take us directly into our muscle structure. Now, when I say muscle structure, I'm referring to the structural organization of muscles. Just as the body has its own structural organization and proteins have their own structural organization, muscles are going to have their own structural levels of organization as well. So in every muscle, we're going to have connective tissue elements that can be found separating the muscle fibers into different bundles and individually. When we break down a muscle cell, we'll find contractile elements and organelles inside of the cell that can further be broken down into their respective parts and proteins. So muscle structure refers to the macroscopic view that we get from our muscles all the way down to the microscopic view. And we see that structure laid out right here in this picture. So I'll just take a quick second to remind you that a muscle cell is referred to as a muscle fiber. So when I say muscle fiber, all I'm referring to is a cylindrically shaped, very long muscle cell. Now, if we have a bundle of these muscle fibers, we're going to call that a fascicle. And we'll see fascicles as part of the structural organization of muscles. So let's start from the macroscopic scale and work our way down. So we can see that this is showing us what a muscle would look like. This very well could be any muscle that you prefer. I'll use the biceps brachii as an example because I've already talked briefly about that muscle. Every muscle is going to be attached to bones by some type of tendon. And around the outside of our muscle, we're going to have a connective tissue layer known as the epimysium. Epi means upon, and my means muscle. So this is the connective tissue that lies upon the muscle. Now every muscle, if we break down muscle into its smaller groups, we're going to get little bundles of muscle fibers known as fascicles. And so fascicles can be shown right here. This is going to be a fascicle, which is just a bundle of muscle fibers that have been wrapped up tight in their own connective tissue layer. This connective tissue layer is referred to as the perimysium. So the epimysium wraps around the entire muscle itself. The perimysium is going to be our connective tissue layer that wraps around these bundles of muscle fibers known as fascicles. And every fascicle is made up of muscle fibers as well as additional connective tissue known as endomysium. So each individual muscle fiber is going to get its own wrapping of connective tissue as well, and that is referred to as the endomysium. So there's 3 layers of connective tissue: the epimysium, the perimysium, and the endomysium, and I will discuss these further on the next slide. But the structural organization of muscle does not stop at the muscle fiber. We can further break down the components inside the muscle cell itself. So I've constantly mentioned that muscle tissue is specialized because it has a very special organelle that allows it to contract. So inside of our muscle cell, we have lots of more cylindrically shaped structures known as myofibrils. Myofibrils are the structure that allow for muscle contraction. These are the contractile elements of muscle cells that give them the ability to contract as they do. So you can see that we have one muscle that's made up of a bunch of bundles of muscle fibers known as fascicles, and every fascicle is made up of individual muscle fibers, and every muscle fiber is going to contain individual myofibrils. So this takes the macroscopic view of muscle and brings it all the way down to the microscopic level. But this is not the end of our structural organization of muscles. We can actually further separate myofibrils into individual repeating units and other protein filaments as well. And we will discuss those shortly. But on this slide, I want to take a second to go over the different types of connective tissues once again that we see wrapping around our muscles. So we have epimysium, perimysium, and endomysium. All of these are different connective tissue elements of muscle that will wrap around muscle tissue and help to protect it. So first and foremost is the epimysium. Epi means upon, so this is going to be the outermost layer of connective tissue, and it's going to be wrapping around the entire muscle itself. So the epimysium is a layer that wraps around the entire muscle. The perimysium is our next deepest layer of connective tissue. This is the connective tissue that wraps around fascicles. Now fascicles, again, are just bundles of muscle fibers. Between 10 and 100 muscle fibers can make up a fascicle. So the perimysium is going to wrap around these individual muscle cells and sort of partition them into different groups. Lastly, within the fascicle, we'll also have a layer of connective tissue that wraps around each individual muscle fiber. So from the muscle fiber up, we have the muscle fiber wrapped in endomysium, and then we're going to take a bunch of those, bundle them up, and then wrap some more perimysium around those, and that will give us a fascicle. And then we're going to take a bunch of fascicles and stick those together, wrap epimysium around them, and that's going to refer to our muscle as a whole. So this slide just gives us another way that we can look at the structural organization of muscle. So you can see the tendon attaching to the bone, and this is going to be continuous with our epimysium, which wraps around the muscle in its entirety. Each muscle is made up of different muscle fascicles that are just bundles of muscle fiber, and each fascicle is wrapped up in perimysium, and we can see the perimysium wrapping around each individual fascicle. Each fascicle is made up of muscle fibers that are wrapped in their very own endomysium. Endomysium will wrap around each individual muscle fiber. And within every single muscle fiber, we'll have contractile organelles known as myofibrils. Now, I really want to just take a second to look at this picture here on the left that was taken with an electron microscope. This actually gives us the ability to see these myofibrils inside of a muscle fiber. So there's lots of different myofibrils that are all oriented along the fiber length. And these organelles are what allow us to contract our muscles forcefully. Now we're going to continue breaking down our muscle cell, but I just want to take a second to show you that there are different protein filaments that can be found making up the myofibril. So our structural organization of muscles does not end at the myofibril, we can take it even further. So here's just an enlarged version of this picture that you guys will be able to use for your study purposes. So let's review what we've learned so far. Each skeletal muscle is going to be made up of different fascicles and is going to be wrapped in a layer of connective tissue known as the epimysium. Fascicles, which we find making up the muscles, are bundles of muscle fibers, and those are going to be wrapped in perimysium. Each muscle fiber gets its own connective tissue wrapping known as the endomysium. And inside of our connective tissue, we're going to contain these individual contractile organelles known as myofibrils. Now, most of this stuff here on this slide we actually have not discussed yet, but we will get there eventually. So just hold on and we will eventually get to the sarcoplasmic reticulum, the transverse tubules, and how muscles are going to contract. So inside muscle fibers, we have our contractile elements, which are known as myofibrils. Now we're going to have lots of myofibrils in every single muscle fiber, but each myofibril is a very long cylinder that's going to stretch in the direction of the skeletal muscle fiber. So each myofibril can further be broken down into structures known as sarcomeres. Sarcomeres are these smaller cylindrical elements that are found attached to each other at the sides, and so myofibrils are these long cylinders, but sarcomeres are just little units that are making up the myofibril themselves. And the sarcomere themselves are going to contain the different myofilaments or muscle filaments that we see allowing for muscle contraction. So we have a few major myofilaments that we'll see that will help to aid in contraction. We'll have the thick filament, which is composed of the myofilament myosin, and we'll have the thin filament, which is composed of other protein filaments known as actin, tropomyosin, and troponin. We're going to discuss each of these proteins in detail when we get to muscle contraction, but I just want to give you an understanding that there are myofilaments that make up every sarcomere. More than one sarcomere is going to make up every myofibril. Our myofibrils are going to be found densely packed inside of our muscle fibers. Muscle fibers make up fascicles, and fascicles make up muscles. So make sure that you take the time that you need to go through and make sure that you have all of these different levels of our understanding of different muscles in order. Now, every muscle fascicle is going to contain parallel bundles of muscle fibers. However, not all fascicles will be arranged in a parallel fashion themselves. Fascicles may be arranged rather irregularly, and depending on the fascicle arrangement, this is going to determine how strong a muscle is or how flexible a muscle is. So the more fascicles that are arranged in parallel, the more strength is going to be coming from our muscle. Just as we saw with dense regular connective tissue, the more fibers we have regularly oriented, the stronger our muscle is going to be. Specific muscles on our bodies need to have this parallel fascicular arrangement. However, other muscles on our body do not necessarily have the function of providing strength and power. As we discussed earlier, some muscles are meant to be stabilizers. So depending on the needs of the muscle, that's going to determine what the fascicle arrangement is. So on this slide, I just want to take a second to introduce and review some terms that we have learned and some terms that we have not heard. But what all of these terms have in common is that we're about to start using them very, very heavily. So let's go over some of these terms that we see. So the sarcolemma refers to the plasma membrane of our muscle cell. Myo means muscle, just as sarco means muscle. So sarco will often refer to flesh. So the sarcolemma of a muscle fiber is its plasma membrane. Now sarcoplasm just refers to the cytoplasm of our muscle cell. Every cell is going to have different cytoplasm, but our muscle fiber's cytoplasm is stuffed with myofibrils. Those are those contractile organelles that we see. So myofibrils are what's actually going to be allowing our muscle cells to contract. Now, every myofibril is made up of repeating smaller cylinders known as sarcomeres. Sarcomeres are the contractile unit of myofibrils. Now, in muscle cells, instead of referring to it as the endoplasmic reticulum, we call this the sarcoplasmic reticulum. The sarcoplasmic reticulum is just the endoplasmic reticulum of our muscle fiber. And I very, very briefly mentioned in Chapter 3 that in muscle fibers, the sarcoplasmic reticulum is storing and releasing calcium. Calcium is a major, major element for muscle contraction and relaxation. And we'll see that the sarcoplasmic reticulum is going to play a major role in what we call excitation-contraction coupling, which is just muscle contraction. And we'll also have myofilaments. These myofilaments, or protein filaments that can be found inside of our myofibrils, are going to eventually help with muscle contraction. So we'll see the thick filament and the thin filament sliding past one another. So myosin is the name of the thick filament, and actin can be found in the thin filament. So every muscle fiber is going to have its plasma membrane known as the sarcolemma, and it's going to have its own cytoplasm known as sarcoplasm. And this sarcoplasm is going to contain myofibrils and sarcoplasmic reticulum. Those are two of the organelles that we see specialized inside of a muscle fiber. One of these is the contractile organelle, and the other is going to store calcium and release it later during muscle contraction. And the myofilaments are going to be making up the myofibrils. These are the smaller protein filaments found within our sarcomeres that are going to slide past one another. And we're going to talk about the sliding filament theory here in just a minute. But on top of showing you all of the connective tissue elements and muscle fibers and so forth again, I just want you guys to get a good idea that we can see the sarcoplasmic reticulum very, very heavily wrapping around our individual myofibrils. So there's lots of sarcoplasmic reticulum found within our muscle fibers. We can also see a myofibril extending outwards here, and we can see these individual parts known as sarcomeres. Sarcomeres are the individual units of the myofibrils made up of the myofilaments. And we can see this thick filament and the thin filament inside of the sarcomere. So on the next slide, we're actually going to discuss the structure of the sarcomere and which protein filaments we can find and where they're at. But on this slide, I just want to show you what a muscle fiber is going to roughly resemble with its myofibrils, sarcomeres, thick and thin filaments, sarcoplasmic reticulum, and the sarcolemma. So we will now discuss the structure of the sarcomere. Every sarcomere is going to have the same exact elements found inside them. And again, sarcomeres just make up the myofibril. So this whole cylinder is the myofibril, and this individual unit found on the cylinder is known as a sarcomere. So this is one way to look at the sarcomere, but we can also look at it through this lens right here. So let's go ahead and get into the individual parts. First and foremost, every sarcomere is going to have a thick filament and a thin filament. The thick filament is this reddish filament right here that has these little heads that stick off, and this is known as myosin. These little heads are known as myosin heads. And the myosin heads are going to bind to the thin filament, which will contain actin. And the thin filament is shown right here in yellow. Now along the center of our sarcomere, we're going to have the M line. I'd like to think that this is the M line that stands for the middle line. So the M line is going to be found right in the middle. On either end of the sarcomere, we're going to have two zigzag structures that are shaped like Z's, and this is known as the Z disc. I also remember the Z disc is found at the ends of the sarcomere because Z is found at the end of the alphabet. We'll also have another filament known as titin, which is an elastic filament, which is going to help our muscles retain its shape after we have contracted. So the thick filament contains myosin, the thin filament contains actin. The thick filament is attached at the M line, which is in the center, and the thin filaments are going to be attached to the Z discs, which are on each end of the sarcomere. And you can see the Z disc is found on either end of the sarcomere. Now at the beginning of this presentation, I explained striations. Striations are stripes. They are the light and dark bands that we see on skeletal and cardiac muscles. And those striations are actually formed by what we know as the A band and the I band. The A band is a structure that runs for the entire length of the thick filament. So the entirety of the myosin proteins that can be found is going to represent the length of the A band. Now the I band is going to be found at the end of the myosin, and it's going to stretch all the way until the myosin of the next sarcomere. So the I band actually spans two different sarcomeres. So the A band is the entire myosin, this is the dark band, and the I band is not going to contain any myosin at all. This is the light band. So those striations that we see in skeletal and cardiac muscle are just the repeating units of these sarcomeres that are showing us the A band and the I band in sequence. So the way I like to keep these in order is that the A band stretches the entirety of the myosin, and it's going to be a little thicker than the I band will. The I band is going to be everything except for myosin, and it's going to be a little thinner. Now I is a much thinner letter than A is, so A is going to be a lot thicker, and that's how we'll remember that the A band is a lot thicker than the I band. Now we have one other zone that I'd like to point out, and that is the H zone. The H zone is found starting at the ends of the thin filament, stretching all the way to the next thin filament. So the H zone is the only location where we have myosin. Now it's important to mention that myosin, the thick filament, and the thin filament, which will contain actin, are actually at some places going to overlap with one another, and we will call those zones of overlap. So the H zone is going to contain the thick filament and the M line, while the A band is going to contain the thick filament, the M line, as well as part of the thin filament as well. And that is due to these zones of overlap. The I band is not going to contain any part of the thick filament at all. It will contain the thin filament, the titin proteins, and the Z disc. So here we just see another explanation of each of these parts of our sarcomere. The Z discs are going to be Z-shaped, and they are going to separate one sarcomere from the next. The A band is going to be the dark band that we see on our striations. This is the middle part of our sarcomere, and it's going to contain the thick filament and part of the thin filaments that are overlapping with the thick filaments. We can very nicely see the A band shown right here. The I band is going to represent the light band that we see on our striations. This is going to contain the thin filament, but no thick filament at all. And the I-band will also contain a Z disc. The H-zone is going to only contain the thick filament, but no thin filaments. The H-zone is found right here, and it will only contain myosin as well as the M-line. And the M-line is the location found right in the middle of our sarcomere that's going to anchor myosin to the sarcomere itself. So I'd like you to feel comfortable with each of these different components of our sarcomere. And I'd also like you to feel comfortable with the different bands that we see, as well as the H-zone. I'd like you to know the limits of each of these bands and what can be contained within each. So as I've mentioned, myosin is found in the thick filament, and actin is going to be the major component of the thin filament. Those are both contractile muscle proteins. Now, as we see in this picture, there's an overlap between the thick filament and the thin filament. And this is very important for us to understand the sliding filament theory. When muscles contract, these two myofilaments are going to slide past one another, and the ends of the thick filament are going to get closer and closer to each Z disc. And ultimately, the length of every sarcomere is going to decrease. And when all of our sarcomeres decrease in length, that's going to produce the shortening of our muscles. So myosin and actin are the contractile proteins because these two proteins will slide past one another. But we'll also have two other major proteins that help to regulate the process of muscle contraction. And these are our regulatory proteins. These are troponin and tropomyosin. Both of these regulatory proteins can actually also be found on the thin filament with actin. So let's go over myosin and then we'll talk about the thin filament. Myosin is the major component of the thick filament. And myosin has a very interesting structure. Myosin sort of looks like two golf clubs that have wrapped around one another very, very tightly. But the myosin heads are very special because they act as an ATPase. This is an enzyme that hydrolyzes ATP and will use the energy on something else. So every myosin head is going to have two binding sites. It's going to have an actin binding site and an ATP binding site. When ATP comes in and binds to this part of the myosin head is going to get energized. It's going to hydrolyze that ATP, and the energy released from ATP is going to energize this myosin head. And that's going to allow our myosin head to bind to the actin proteins. So it's very important that we understand that we have two different binding sites on every myosin head, and the myosin heads act as an ATPase. Now when we look at the thin filament, there's a few more filaments going on here, so let's go over each of these. Actin are these yellow balls that have been chained up together, sort of like a beaded necklace. This is what actin is going to look like, and we have two of these actin strands that are wound together, and that gives us a spiral. This is the major component of the thin filament, is actin. Now on every actin molecule, we're going to have myosin binding sites. And these are going to be the places where myosin is going to bind. So on the myosin head, we have actin binding sites, and on the actin, we're going to have myosin binding sites. But also on the thin filament, we're going to have troponin and tropomyosin. So tropomyosin is this long protein, and it's going to cover up the myosin binding sites on the actin protein. Tropomyosin is actually preventing myosin and actin from binding in this picture. When tropomyosin is covering these myosin binding sites, that's preventing the binding from actin to myosin, and we will have no muscle contraction when tropomyosin is doing this. So tropomyosin is eventually going to get moved out of the way, and that's going to allow for the binding between actin and myosin. But in order for tropomyosin to move out of the way to allow for muscle contraction, we're going to need one more protein known as troponin. So troponin is our other regulatory protein, and when calcium is released from the sarcoplasmic reticulum, that's going to come and bind to troponin, and that's going to induce a conformational change in the shape of troponin. This is not the first time we've seen a conformational change. We also saw that with carrier proteins. So when troponin undergoes its conformational change, it's going to change in shape, and it's going to pivot tropomyosin off of the myosin binding sites. And once tropomyosin has been moved off of the myosin binding sites on actin, that's going to allow the myosin proteins to come in and bind to actin to give us our muscle contraction. So I know that may sound like a lot, so I've got this slide as a summary to explain each of these types of proteins, whether they're regulatory or contractile, and what their major functions are going to be. And then we're going to go over these events on the next couple slides in different pictures that will show us cross-bridge cycling and excitation-contraction coupling. So in summary for what we discussed on the last slide, myosin is the major protein that makes up the thick filament. This is going to have some tails that are wrapping around one another and two myosin heads. And there are two binding sites on every myosin head, an ATP binding site and an actin binding site. Remember, the actin binding site is on the myosin molecule. The myosin heads at their actin binding sites are going to bind to the actin molecules at their myosin binding sites. And when myosin binds to actin, that's going to allow those filaments to slide past one another and produce what's known as muscle contraction. Now, actin is the other contractile protein that we see, and this is the major component of our thin filament. Each actin molecule is going to have a myosin binding site, whereas myosin had the actin binding site. So don't get those mixed up. So during muscle contraction, the actin binding site on myosin is going to be bound to the myosin binding site on actin. And that binding is only facilitated by ATP coming into the myosin head and calcium coming in to bind to troponin, where it can move tropomyosin out of the way. So let's just real quickly discuss tropomyosin and troponin once more. Both of these are regulatory proteins. Tropomyosin and troponin are both regulatory proteins, and they are both going to be found also on the thin filament with actin. Now, tropomyosin specifically is going to be sitting on the myosin binding sites on actin when muscle is relaxed. So we've got two stages, muscle relaxation and muscle contraction. During muscle relaxation, tropomyosin is covering up those myosin binding sites and preventing actin and myosin from binding to each other. And therefore, we will have no sliding of filaments and therefore no muscle contraction. However, whenever troponin is in play and calcium ions are present, calcium is going to induce a conformational shape change in troponin. And when troponin changes its shape, that's going to pivot tropomyosin off of those myosin binding sites on the actin molecules. Now, troponin, remember, is attached to tropomyosin. So when calcium comes in and binds to troponin and this changes its shape, the shape change is what forces tropomyosin off of those binding sites. And once tropomyosin is no longer covering the myosin binding sites on actin, that will allow for myosin to come in and bind to actin, and those myosin heads can walk up the various binding sites on the actin molecules. So the way that this is going to work is these myosin heads are going to bind to those binding sites when tropomyosin's out of the way, and they're going to walk up. We'll depict this as head number 1, and we'll depict this as head number 2. So head number 1 might bind right here, and then head 2 is going to bind here. And once head 2 binds here, head 1's going to detach, and it's going to walk its way up to this one. So head 1 is going to jump from binding site to binding site. And once 1 has bound, 2 will release and then eventually walk its way up further. So we have this walking effect where the two myosin heads are walking up the actin molecule on the myosin binding sites of actin. I definitely want you to know the functions of each of these proteins and the characteristics of each. Now that is going to take us into our next topic, which is the contraction and relaxation of our skeletal muscles. This brings in two new concepts known as cross-bridge cycling and excitation-contraction coupling. Cross-bridge cycling can also be referred to as the contraction cycle and refers to the action of myosin heads walking up the actin molecules. And we'll see the contraction cycle on the next slide. Excitation-contraction coupling is where we link this cross-bridge cycling with a nervous action potential sending a signal to our muscle fiber telling it to contract. So let's take a look at cross-bridge cycling, and then we will get into excitation-contraction coupling. So first and foremost, I'll say again about cross-bridge cycling is that this is the process by which myosin heads are going to bind to the actin and walk up actin. When they walk up actin molecules, that's going to pull the two Z discs closer towards the M line. And overall, that's going to produce a shortening of our muscle, and that's referred to as muscle contraction. So every time our muscles contract, we have this contraction cycle taking place. So there are 4 steps in the contraction cycle. The first step is ATP hydrolysis. The second step is cross-bridge formation. The third step is the power stroke. And the fourth step is the detachment of myosin from actin. So two other quick notes I want to mention before we get into this contraction cycle is that in order for contraction to happen, calcium must be present and bound to troponin. We can see that troponin is depicted by the three blue circles, calcium is depicted by the purple circle, and now because calcium and troponin have come together, tropomyosin is no longer covering these binding sites on actin. So if actin's binding sites are open, we can have this cross-bridge cycling. This is only allowed when calcium is present. And the second thing I want to remind you of is that the myosin head has two binding sites, one for actin and one for ATP. So cross-bridge cycling is going to require ATP. So ATP is going to come into this ATPase, which is the myosin head, and the myosin head is going to hydrolyze that ATP. It's going to cut off that phosphate group and become energized. And when this becomes energized, we refer to the myosin head as cocked because it's ready to bind to actin. So in our first step, ATP is hydrolyzed and that energizes our myosin heads. Now in the second step, we call cross-bridge formation because this is what cross-bridge cycling refers to. We form a cross-bridge connecting the myosin molecule to the actin molecule, and this is what is known as a cross-bridge, hence why we call it cross-bridge cycling. Now after this step occurs, we're going to release the phosphate group, and that's going to leave the ADP still attached to the myosin head. And I'd like you to be aware that ADP is actually going to be attached to the myosin head for most of this whole process. So the third step is the power stroke. The power stroke refers to the actual movement of our myosin heads and the sliding of the filaments. So myosin is attaching onto these binding sites on actin and pulling actin past. You can see that actin is showing us that it's moving in this direction. Actin is moving past myosin because these myosin heads are pulling actin in this way. So a good way to say this would be that myosin is pulling the two thin filaments towards the M line. And the two thin filaments are getting closer and closer to each other to almost touching at the M line at full contraction. Our last step is the detachment of myosin from actin. Now there's a very important requirement to this step, and that is ATP. ATP must bind to the myosin head in order for the myosin heads to release from actin. If ATP is not present, we do not get this last step. And this is actually clinically why we see a condition known as rigor mortis. After death, muscles are going to lock up, and that is because there's no ATP present to come and release the myosin heads from the actin. So with the lack of ATP, we're not going to get the detachment of myosin from actin. And this is a requirement for the fourth step. So the detachment of myosin from actin requires ATP. If ATP is not present, myosin and actin will remain attached to one another. So in summary, for cross-bridge cycling, we have ATP hydrolysis, where we energize the myosin heads. In step 2, we're forming a cross-bridge, where the actin binding site on myosin is going to bind to the myosin binding site on actin. In the third step, we have the power stroke, which is the actual true muscle contraction. We're actually shortening the length of our muscle with the power stroke. You can actually see the angle by which we are drawing here is actually a little less. This is now a 45-degree angle, and up here it was a 90-degree angle. This is showing us that the actin molecule is actually moving closer towards the M line. And as this last step is requiring ATP, this is the detachment of myosin from actin. So this process will continue to go in a circle. As long as ATP is present to detach myosin from actin, once ATP detaches myosin from actin, other cross bridges are going to form at different places on the actin molecule. And that's going to continually pull the actin molecules towards the M line. And I'll just give you one more reminder that, of course, calcium is going to be required for this cross-bridge cycling or the contraction cycle to occur. Okay, so this slide is going to discuss excitation-contraction coupling. This is our big concept for Chapter 10. Excitation-contraction coupling refers to combining the events that occur with a muscle action potential, sending a signal to a muscle fiber to allow for muscle contraction. So these are the events that occur every single time you contract or relax any of your muscles. Now on this slide we're going to discuss what happens inside of the muscle fiber, and on the next slide we're going to discuss what happens inside of the nerve fiber. So on the right side we see relaxation, and on the left side we see contraction. So we're going to start with relaxation and describe this picture and all of the things that are going on. Now, there are lots of components that can be found inside a muscle cell. Some of the components that we're already more familiar with are the myofibril and the sarcolemma, which is the plasma membrane, as well as our sarcoplasmic reticulum, which is shown here in yellow with these purple dots in it. And as I've described on the other slide, calcium is representing these purple dots. Now, that being said, there are some invaginations inside of the plasma membrane, and there are also some membrane proteins that we need to introduce also. So in the sarcolemma of the muscle fiber, our sarcolemma is going to bend inwards into our cell here. And there's going to be various inward bendings or invaginations of our sarcolemma. These inward foldings are referred to as transverse tubules or T-tubules. And inside every T-tubule are going to be certain membrane proteins. So this blue one that has the 4 little subunits on it is the voltage-gated calcium channel. There are 4 voltage-gated calcium channels that can be found embedded in the sarcolemma. And we'll also have this green membrane protein that's embedded in the membrane of the sarcoplasmic reticulum. These are referred to as the calcium release channels. So the calcium release channels are going to be the channels that release calcium from the sarcoplasmic reticulum. So in the sarcolemma, we have these invaginations known as T-tubules, and then embedded in the membrane of our sarcolemma, we will find our voltage-gated calcium channels, and then embedded in the membrane of the sarcoplasmic reticulum, we're going to find our calcium release channels. It would be in your best interest to know which of these protein channels can be found embedded in the sarcolemma and which can be found in the sarcoplasmic reticulum. All of these components are necessary for us to fully understand muscle contraction. So at a time of muscle relaxation, the calcium release channels are plugged up by the voltage-gated calcium channels. When the calcium release channels are plugged, all of the calcium inside of our muscle cell is going to be found inside of this sarcoplasmic reticulum. Remember, the sarcoplasmic reticulum stores calcium. So if no calcium is out inside of our cytoplasm and it's all locked up inside the SR, there's going to be no calcium binding to troponin, and tropomyosin will be covering the myosin binding sites on actin, therefore preventing any binding of myosin to actin. This is what happens during muscle relaxation. Now, during muscle contraction, some things are going to change, and that's going to cause a cascade of events that will occur to give us muscle contraction. So the first thing that's going to happen inside of our muscle fiber is we're going to have this muscle action potential. Now, a muscle action potential is firing through our muscle cell, and it's propagating along the membrane. Now, I've mentioned that I'll discuss action potentials in later slides, and so right now I just want you to understand that this action potential is what's causing all of these events to occur. But when this action potential comes through here, this is going to change certain characteristics about the membrane. And this action potential is propagating along the membrane. So this action potential is sensed by the voltage-gated calcium channels. The voltage channels are going to sense a change in the voltage of our sarcolemma. And when these voltage-gated channels sense this change, they're going to dissociate from the calcium release channels that we will see. So over here in muscle relaxation, the voltage-gated calcium channels were plugging up the calcium release channels. And right here we see that these are now dissociated. So the calcium release channels simply are going to release calcium from the sarcoplasmic reticulum. Calcium is going to flow along its gradient from high concentration to low concentration out into our muscle fiber. And calcium is going to go all around our myofibrils. When calcium is in close proximity to our myofibrils, it's going to come in and it's going to bind to troponin. When troponin gets calcium, we know from previous slides, it's going to undergo a conformational change, and that's going to move tropomyosin off of the myosin binding sites on actin. And when tropomyosin is out of the way, the myosin heads can come in and bind to the actin. And this is where we have cross-bridge cycling taking place. When calcium is being released from our sarcoplasmic reticulum, we can have cross-bridge cycling. So once the calcium comes over here and binds to troponin, we're going to see this happen over and over and over again until we stop muscle contraction. So starting with the muscle action potential that propagates along the cell membrane, that's going to get sensed by our voltage-gated calcium channels, which will then cause a dissociation with the calcium release channels. The calcium release channels embedded in the membrane of the sarcoplasmic reticulum will release all of the calcium, and this calcium is going to flow around our sarcomeres. The calcium will eventually come in and bind to troponin, which will undergo a conformational change to move tropomyosin off of the myosin binding sites on actin, which allows for cross-bridge cycling to take place. So again, this will continue until we're ready to relax our muscles. Now, when we're ready to undergo muscle relaxation, we're just going to stop firing this action potential. When this muscle action potential stops firing, we're going to sense this with our voltage-gated calcium channels. The voltage-gated calcium channels will then reassociate with the calcium release channels, just as we saw here during muscle relaxation. So that will put most things back to normal inside of our cell, with the exception of the fact that calcium is now floating around our myofibrils. We have to have a way to get calcium back into the sarcoplasmic reticulum, because if we don't, we're going to leave calcium in close proximity with troponin, and that will continue the process of muscle contraction. So there's one other major element that we need to look at here, and that is the calcium ATPase pump. The calcium ATPase pump is also going to be embedded in the membrane of the sarcoplasmic reticulum. This pump uses ATP to actively pump calcium against its concentration gradient back into the sarcoplasmic reticulum. And once these channels close, the calcium ATPase is going to pump most of the calcium back into the sarcoplasmic reticulum, and that will help to sequester calcium. Now, there's another protein that I'd like you all to be aware of, which is known as calsequestrin. This is a protein that hangs out inside of the sarcoplasmic reticulum, and it's going to bind to calcium and help to package calcium very nicely inside of the sarcoplasmic reticulum so that we can fit more calcium into the SR and store it there more readily. So both calcium ATPase pump and calsequestrin are going to help to get more and more calcium back in to our sarcoplasmic reticulum. Now, there's one other characteristic that I really want you guys to understand about the calcium ATPase pump. The calcium ATPase pump is what is referred to as constitutively active, which means that no matter the case, this is always going to be pumping calcium back into the sarcoplasmic reticulum. So when we go back to our muscle relaxation, the calcium ATPase pump will have pumped all of this calcium back into the sarcoplasmic reticulum. However, during muscle contraction, the calcium ATPase pump is also pumping calcium back into the sarcoplasmic reticulum as well. However, once it goes in, if these remain open, calcium is going to be allowed to flow right back out and go and interact with our muscles. So this is always going to be pumping. The calcium ATPase is constitutively active, and during muscle contraction and muscle relaxation, we will always have the calcium ATPase pumping calcium in to the sarcoplasmic reticulum. Again, this doesn't really matter when we're talking about contraction, because eventually that calcium will just flow right back out. So even though it is pumping in during contraction, it's just going to flow right back out of these calcium release channels and go and bind to troponin. So the major control factor for what caused all of these events to happen is a muscle action potential. So the action potential is what causes muscle contraction, and when that stops, we're going to get muscle relaxation once again. So now we're going to discuss a little bit about action potentials and explain a few concepts about the neuromuscular junction, which is where nerve cells and muscle cells meet. So a quick little review from the one nervous tissue slide we covered in Chapter 4 is that neurons are going to be the main communicative cells of the nervous system. Every neuron is going to have a cell body, dendrites that receive signals, and a very long axon that's going to send out signals. And at the end of our axon, we're going to branch into little axon terminals. Since the axon is where these signals are sent out from our neuron, this is where our action potential is going to propagate. Action potentials are fired down the axon, and those are eventually going to make their way into muscle cells that are attached to that neuron. So we have many different types of neurons that can be found in the body, but specifically the type of motor neuron that's going to be attached to muscle fibers is known as a somatic motor neuron. Motor neurons control muscles. So the neuromuscular junction is the place where a somatic motor neuron is going to come in and meet with a muscle fiber. And the location where the somatic motor neuron and the muscle fiber are going to meet is referred to as a synapse. The location where any neuron attaches with any other cell is referred to as a synapse. So a synapse is just a location where we see the axon terminal of our somatic motor neuron attaching to our muscle fiber. Now we're going to discuss a lot more about synapses in this class, but specifically, the synapse between a somatic motor neuron and a muscle fiber is referred to as the neuromuscular junction, because we have a nerve cell meeting with a muscle cell. So the location where any neuron is going to meet with any other cell is referred to as a synapse. But the neuromuscular junction is a special synapse that specifically denotes all of the attachments between somatic motor neurons and muscle fibers. So on this slide, we're showing you a synapse. This is the axon terminal coming out of the axon of our neuron, and this is representing the muscle fiber. So this is the sarcolemma right here. And at every synapse, we're going to have a tiny little space that can be found between our two cells. And this space is referred to as the synaptic cleft. That's the little space in between our somatic motor neuron and the muscle fiber. Now there's also going to be lots of different channels and receptors that can be found in the synapse. And so certain chemical messengers, known as neurotransmitters, are going to be released into the synapse, and those are going to help to affect the target cell. So that muscle action potential we saw propagating on the slide for excitation-contraction coupling is started here at the synaptic cleft. So there are lots of neurotransmitters that we're going to discuss within the body. And again, we'll touch on those a little bit more in Chapter 12. But specifically in muscles, the main neurotransmitter is referring to acetylcholine. This is the major neurotransmitter that we have in our muscular system that's going to activate our skeletal muscles. So our neuron is going to have little vesicles containing acetylcholine, that neurotransmitter. And what's going to happen is that when the nerve action potential comes down, or the nerve impulse comes down to the axon terminal, that is going to activate voltage-gated calcium channels found embedded in the membrane of the axon terminal. This is going to allow calcium to flow into the cell, which will eventually act on these vesicles to allow them to be exocytosed into the synaptic cleft. So these vesicles containing acetylcholine are going to fuse with the membrane of our axon terminal and release their acetylcholine into this synaptic cleft. When acetylcholine has been exocytosed into the synaptic cleft, this is going to allow acetylcholine to interact with certain receptors, which will affect different channels that can be found in the postsynaptic membrane, which can also be called the sarcolemma in this example. So at every synapse, we're going to have certain channels that are going to interact with these neurotransmitters that have been released. And these neurotransmitters are going to cause the opening of these channels. These channels are ligand-gated channels. Now, a brief review on ligands is any type of chemical that binds to a receptor. So we'll have acetylcholine receptors, and the ligand is going to be acetylcholine. So whatever the neurotransmitter is at our synapse, it's going to affect these channels, which are going to affect the permeability of those channels. You can see that these specifically are ligand-gated sodium channels. So when acetylcholine is exocytosed and interacts with these different ligand-gated sodium channels, that's going to open these channels and allow sodium to flow into our cell. The sodium influx into our cell is what the action potential is referring to. So that muscle action potential we saw that allowed for muscle contraction is all caused by this sodium entering into the cell. Now I know this slide has a lot of words on it, but this slide is essentially explaining everything I just said on the last slide. So we're going to go through this again over here, because I know that this is a big process. So it's all going to start with voltage-gated calcium channels being stimulated by some type of action potential coming from our neuron. When these voltage-gated calcium channels sense this action potential, they're going to open up and allow calcium to flow into the cell. Calcium is going to cause the exocytosis of acetylcholine that can be found in vesicles, and that acetylcholine is going to be released into the synaptic cleft, which is the space between the axon terminal and our muscle fiber. Acetylcholine is eventually going to go bind to ligand-gated sodium channels, and those are going to be found on the sarcolemma itself. When acetylcholine activates these ligand-gated channels, they're going to open up and allow sodium to flow into the cell. And when sodium flows into the cell, this is going to send a signal across the membrane known as a depolarization, which will eventually affect the voltage-gated calcium channels that can be found in the sarcolemma, to eventually lead to the dissociation of our voltage-gated calcium channels from our calcium release channels. We'll then get release of calcium, and that will then go bind to troponin and allow for cross-bridge cycling. So those are all the events that we discussed on this slide. So this is the picture version, and on the next slide you have the word version. Now there's one other thing that I'd like to mention about the neuromuscular junction, and it's that each muscle fiber is going to have only one neuromuscular junction. So what we'll come to see is that neurons are going to branch out and have lots of different axon terminals. Each nerve's axon is going to produce a lot of axon terminals, and each axon terminal will bind with its own muscle fiber. So one neuron can be bound to many different muscle fibers, but each muscle fiber is only going to be bound to one neuron. So this is just another picture to show what's been going on here. We have calcium being influxed into our axon terminal. This is going to cause the exocytosis of the synaptic vesicles containing acetylcholine, which will then be released into the synaptic cleft. The acetylcholine will bind to these ligand-gated sodium channels, and sodium will then flow into the muscle cell. You can see a closer version of this showing the synaptic cleft with acetylcholine being released in here, and then acetylcholine being bound to these different ligand-gated sodium channels, and that's going to allow for sodium to flow into our cell. So to fully summarize the process of excitation-contraction coupling, I have this slide. This slide shows all of the processes that we've discussed in the last few slides, including cross-bridge cycling, muscle contraction, and the action potential propagation. So we're going to start with the action potential propagation. A nerve action potential is fired down the axon, and that's going to stimulate voltage-gated calcium channels that can be found in our axon terminal's membrane. When that allows calcium into the axon terminal, that will cause the exocytosis of these synaptic vesicles containing acetylcholine. Acetylcholine is going to be released into the synaptic cleft. When acetylcholine has been released into the synaptic cleft, it's going to bind to these ligand-gated sodium channels. Ligand-gated sodium channels are going to have receptors for acetylcholine, and that's why acetylcholine is the ligand to allow for those sodium channels to open. When sodium channels open and sodium flows into our cell, this causes what is known as a depolarization. And this depolarization is felt all throughout the cell membrane. So this is what's going to then trigger that muscle action potential that will come by and eventually be sensed by the voltage-gated calcium channels inside of the T-tubule embedded in the sarcolemma. When these voltage-gated calcium channels sense this muscle action potential, they're going to dissociate from the calcium release channels. Calcium inside of the sarcoplasmic reticulum, which is existing in a very high concentration, is going to flow out of the calcium release channels and eventually make its way over to troponin, which can be found on the thin filament. When calcium binds to troponin, this is going to cause a conformational change, and that will move tropomyosin off of the myosin binding sites on actin. And when the myosin binding sites on actin are free, that's going to allow for cross-bridge cycling to occur. And we can actually see that shown in this picture, that the thin filaments are almost touching the M line because the myosin filament is pulling those two actin filaments towards the center, and this is causing a shortening of our muscle, which is known as a muscle contraction. This is going to continue until we stop this nerve action potential. And once this stops, that's going to stop the influx of calcium, which will stop the exocytosis of acetylcholine, which will eventually stop the influx of sodium. If sodium is not flowing into the cell, that's not going to be sensed by these voltage-gated calcium channels, and then these are going to reassociate and close, and no more calcium will be able to leave the sarcoplasmic reticulum. Then, to restore the concentration of calcium in this sarcoplasmic reticulum, we're going to use the calcium ATPase pumps that are always pumping, those are constitutively active, to bring calcium back into the sarcoplasmic reticulum. And when all the calcium has entered back into the sarcoplasmic reticulum, troponin will eventually lose that calcium, and that's going to change its shape back to allow for tropomyosin to cover these myosin binding sites on the actin. Calsequestrin inside of the sarcoplasmic reticulum is going to help organize and hold more calcium so that we can fit more calcium into the SR. And all of these events will eventually occur to relaxation. So what controls this process is whether or not there's a nerve action potential or not. If there's an action potential, we're going to get contraction, and when we want to turn off that nerve action potential, that's going to give us muscle relaxation. Now there's one other enzyme that I'd like to introduce to you that can be found inside of the synaptic cleft, and it is known as acetylcholinesterase, and we see that shown right here. Acetylcholinesterase is an enzyme that is found sitting inside of the synaptic cleft, and this is an enzyme that breaks down acetylcholine that has been released into the synaptic cleft. If we did not have this enzyme breaking down acetylcholine inside of the synaptic cleft, muscle contraction would just continue and continue as long as acetylcholine can be found in the synaptic cleft. So this enzyme is a way by which we can guarantee our muscles are contracting when we want them to. If we want our muscles to contract, we're going to continue to fire nerve action potentials that will release acetylcholine into the synaptic cleft. And acetylcholine is going to be breaking this down, and so we'll need to release more and more acetylcholine if we want to continue contraction. However, when we want to stop contracting, any acetylcholine that is left in the synaptic cleft is going to get broken down by acetylcholinesterase, and that's going to stop the influx of sodium into our muscle fiber. So this is another major enzyme that I'd really like you to be comfortable with, is acetylcholinesterase. We can see that hanging out inside the synaptic cleft in this picture. So that is the overall summary of excitation-contraction coupling. I know this process can seem overwhelming at times, so if you do have any questions about this process, by all means, feel free to reach out and send me an email. I'd be happy to help. We will come back for part 2 of our Chapter 10 lecture video, where we're going to discuss motor units, muscle metabolism, and different types of muscle fibers.